that is comprised of stem cells, at least in some tissues, including the brain, lose that self-renewal competence. And in the brain, the cells, after they leave the ventricular wall, actually retain what we call multilineage competence. They can still make glia, astrocytes and oligodendrocytes, and at least under appropriate conditions can still make neurons. But what they cannot do is keep dividing. So they lose self-renewal competence. And so in the human, the progenitor population that's derived from the stem cell largely persists within brain parenchyma. And so from my own standpoint, over the years, as we initially were looking at neural stem cells as a source of neurons, I became really enamored of the glial progenitors that derive from those neural stem cells, because I realized that in humans at least, that's by far the major progenitor cell population. The neural stem cell is, again, relatively scarce. The neurogenesis itself, the production of neurons from those neural stem cells in humans, is a relatively rare event, we think now. It persists within the hippocampus. Even there, the numbers aren't really clear, but it persists within the adult hippocampus. Early in postnatal development in the cerebellum and the cerebellar granular layers, perhaps in the olfactory bulb, also in a manner that probably degrades with age. And so humans, as far as is known now, we don't have that many instances of ongoing neuronal production. And so the stem cells are not all that active from the standpoint of neuronal production in humans. Again, there are isolated areas where it still occurs, and the kinetics still aren't really well understood. But what's really clear is that there are tremendous numbers of glial progenitors that are produced from those neural stem cells that persist within the adult and continue to be produced and turn over within the adult, meaning more of them are always, some of them are always dividing and more of them are always, over the years I became more and more interested in that population, just because it looked to be much more important in human biology. And from my own biases, I'm still a clinician, and so human biology, as opposed to that of lower species, has some specific interest. And so I started focusing more and more on glial biology as time went on, and that morphed into looking at the role of glia in disease, and that in turn morphed into trying to model the roles of human glia in different human diseases.
Yeah, and I sort of feel bad for you in the sense that neurons come first in development. So trying to make glia from stem cells, embryonic ones at least, takes a long time traditionally. And some of these protocols, they're like day 60, day 90. And if, you get a little bit of contamination, you lose a quarter of the year's work. And so, talk about trying to produce. I know your lab has used several great protocols using PDGF receptor to sort and purify, say, oligoprogenitors. What's the state of, have you guys been able to push the production of the cells to a faster timeline, or are we still stuck with day 60 or day 90 protocols?
We're still stuck with